Imagine you or a loved one were diagnosed with brain cancer. It's probably glioblastoma, or GBM, the most common type of brain tumor. What would your options be? Unfortunately, even after decades of research, the tools we have to treat GBM remain extremely limited. Even with the full current standard of care, which is surgery followed by chemo and radiotherapy, median survival is only about 14 months, and the tumor almost always comes back. This extremely aggressive disease causes about 10,000 deaths in the United States each year. So why is GBM so difficult to treat? For one, the tumor grows in the protected environment of the brain, where physiological barriers, such as the blood-brain barrier, carefully filter what goes in or out. Additionally, GBM is highly invasive, growing deep into the surrounding brain tissue and making it difficult to remove entirely in surgery. GBM is also extremely sneaky, using a variety of mechanisms to avoid detection and destruction by the immune system. Okay, so what can we do? The goal of my project is to create a new treatment that can address these concerns, to help fully eliminate the original brain tumor and teach the immune system to better identify and fight GBM so the tumor cannot return. And the key component for this therapy comes from the tumor cells themselves. See, almost every cell type in the body secretes small particles called extracellular vesicles, or EVs. As a natural cargo delivery system, they can carry a wide array of molecules. They can cross physiological barriers, like the blood-brain barrier, and they have shown limited toxicity to cells. Furthermore, EVs collected from tumor cells have additional useful characteristics. For one, they contain lots of tumor antigens, which can help activate the immune system against tumor cells. They also have shown homing abilities to tumor cells, which may help improve the specificity of the treatment delivery. My project aims to take these so-called Trojan horses of drug delivery and arm them with a tumor-killing cytokine and a cancer vaccine that the vesicles can deliver to the tumor. The killing cytokine is presented on the surface of the EVs, and it can induce apoptosis in tumor cells to destroy them. The cancer vaccine contains short peptide sequences of 3 GBM-associated antigens that are common in both mouse and human GBM tumor cells. I hypothesize the antigens will be taken up by special immune cells called dendritic cells, which can present the antigens to other immune cells called T cells and activate them to kill tumor cells. With this two-pronged approach, I aim to develop a treatment to help eradicate the initial tumor and prime the immune system against tumor recurrence. First, I am assessing these types of vesicles separately on the bench and in mouse experiments. For the killer EVs, I am testing their killing ability by treating tumor cells with different concentrations of EVs for several days before measuring tumor cell survival. Later, I plan to establish tumors in mice and see if treating with killer EVs can reduce the size of the tumors and improve mouse survival. For the vaccine EVs, I will be testing their ability to activate T cells against tumor cells by treating naive immune cells with the vaccine EVs and then seeing if those immune cells were activated to kill tumor cells. Later on, I will test if treating mice with vaccine EVs before trying to implant tumor cells can help the mice prevent the tumors from establishing. After testing both of these therapies separately, I plan to combine them to make a therapy that can both kill tumor cells directly and activate an immune response against GBM. With this project, I hope to help work towards better treatment options for glioblastoma that can give patients more hope, so that in the future, a brain cancer diagnosis won't have to be so scary. Thank you.